In the last lesson, we spoke about the two different types of digestion that work together to break down food into smaller pieces that our bodies can absorb. The first was mechanical digestion, which begins in the mouth. Our teeth and jaws work together to crush and grind food into smaller bits. We also discussed chemical digestion. This also begins in the mouth with enzymes present in our saliva. For example, amylase is the enzyme in our saliva that helps to chemically break down carbohydrates into smaller sugar molecules. So, we know enzymes help to chemically break down molecules into smaller ones, but what exactly are they? Enzymes are made up of proteins. They are also known as catalysts. Catalysts help to speed up chemical reactions. Enzymes are specific, meaning they can only break down certain molecules. For example, amylase can only break down carbohydrates and not lipids or proteins. Most enzymes also end in the letters ASE, so it is easy to identify which is the enzyme and which is the molecule that it's breaking down. There are two basic groups of enzymes. There are building enzymes, which synthesize or make large, complex molecules from smaller ones. However, for our purposes of studying the digestive system, we will focus only on the enzymes that break down large molecules into smaller ones. These are called digestive enzymes. And as you can see from this figure at the top, in a synthesis reaction, an enzyme works to bring together two smaller molecules to make a larger one. And a digestive enzyme takes one large molecule and breaks it down into smaller pieces. Enzymes are special proteins that help aid in the chemical reaction of a specific molecule. Each enzyme has a specific shape. Its shape allows it to take part in only one kind of chemical reaction. Enzymes are named for the reactions they help, so it is easy to identify the molecule that they break down. For example, proteases break down proteins and lipases break down lipids. Note the spelling of protease, lipase, and sucrase. They all end with an ASE, so you can immediately know sucrase is the enzyme and sucrose is the molecule that's being broken down. Enzymes cannot be used up. They are reused over and over again for the same reaction. The active site of the enzyme recognizes the substrate, or molecule that's being broken down, by its shape. Each enzyme has a specific shape which recognizes the shape of the food molecule, whether it be a carbohydrate, protein, or lipid, allowing the enzyme to break it down. The shape allows it to take part in only one type of reaction. You can think of this system as a lock and key. A lock can only be opened up by one specific key that has the correct shape to fit into the lock. In this analogy, the lock is the enzyme and the key is the substrate that fits into it. In the first step, the substrate recognizes the enzyme shape and binds to it. In the second step, the substrate is converted to its products or broken down into smaller pieces. This would be like carbohydrates being broken down by amylase into smaller sugar molecules. The products are then released, so the active site of the enzyme is now open and ready to be used again in the same chemical reaction. So, we just learned a lot of new things. Let's do a quick review. Enzymes help speed up chemical reactions. Their role in the digestive system is to help break down large molecules into smaller ones that our bodies can use to grow, repair, and fuel daily activities. A substrate is the molecule that the enzyme breaks down. This would either be a carbohydrate, protein, or a lipid. The enzyme-substrate complex is what is formed when the enzyme and molecule are joined together. The active site is the part of the enzyme that the substrate molecule fits into. Okay, well, I hope this lesson has helped give you a better idea of what an enzyme is and their role in the digestive system. In the next video, we will review the 3 enzymes that break down carbohydrates, lipids, and proteins, and in which parts of the digestive system they occur.